Hi, today we're going to look at the structure of DNA and what components the DNA is made of and what scientists contributed to the structure of DNA. So, first of all, we need to know what DNA stands for, and it stands for deoxyribonucleic acid. DNA is where all of the cell's information is contained, and the one important information that's contained is that it makes proteins, and so it holds all the genetic information in order to make the proteins. It actually makes mRNA, which is messenger RNA, and all of this is happening in the nucleus. And then the messenger RNA will go out into the ribosomal RNA and make your proteins. So basically, a DNA strand is a macromolecule, and we have studied macromolecules before, and in particular, it's a nucleic acid. And so we've talked about what nucleic acids, so go back to your memory bank and remember those, but I'll give you a little bit of information as a reminder. So they're made up of monomers, and the monomer that makes up a nucleic acid is called a nucleotide. And the nucleotide structure is at the bottom on the right hand side. It's composed of a phosphate group, a sugar, and a base. The sugar in DNA is called deoxyribose, and then you have four bases. You have adenine, thymine, guanine, and cytosine as your four bases. So those four bases are composed of two different types, the purine or the pyrimidine. The purines have two rings that are connected together, and the pyrimidines only have one ring that's connected together. They also usually use the symbols for these bases and not the whole name. So thymine is a T, adenine is an A, cytosine is a C, and guanine is a G. And here are the structures where you can see them a little bit better. So adenine and guanine are purines, and cytosine and thymine are pyrimidines. So we're going to watch a little video about DNA that's going to give us a little bit more information.
On our planet, and perhaps countless others, life arose. The DNA molecule, which is the basis of all organic life on Earth, is more intricate by far than any spiral galaxy. Because the structure of DNA contains something new, something that was missing from inanimate matter before the origin of life. It contains information. The DNA molecule encodes not only the information necessary to make copies of itself, but the information necessary to construct an entire organism. The blueprints for an ant, or a dolphin, or a bullfrog, or even a person. All of this information is somehow built into the structure of an organism's DNA in a molecular code billions of letters long. Each of your chromosomes is a single continuous strand of DNA, one enormous molecule. And as the molecule winds or unwinds, each chromosome can vary in shape, from a stringy open formation, if it is performing a task, to a remarkably tight-packed mass if the cell is preparing to subdivide. This winding is so efficient that it packs 3 feet of DNA helix into a cell nucleus. This helix is 3.4 angstroms per step. This particular clump of DNA is chromosome 17, magnified some 50,000 times. And as we zoom in, you can begin to make out the rungs on the ladder. Each of these rungs is just one of the 3 billion ladders that make up the book of our instruction manual. These steps of the DNA ladder are composed of just 4 different molecules: adenine, thymine, cytosine, and guanine. And it is common to refer to them by their initials: A, T, C, G. There are two of these base molecules for each step on the ladder, and it is always either A and T together or G and C together. So if you know one side of the step on the ladder, you automatically know the other side as well. Step by step, and 3 billion letters long, this is the formula for a human being. The existence of this tiny code, written molecule by molecule inside each of our cells, is a remarkable discovery all by itself. But scientists have now read that code, examined it letter by letter, and written it down.
So, the structure of DNA was a collaborative effort. There was not just one scientist that figured out how DNA worked or what the structure of DNA looked like. And so, this started back in the early 1900s, this gathering of information. So, at that point, they knew that cells passed information from cell to cell. And they basically didn't really know what was carrying that information. Some of the scientists thought that it was a protein that was responsible for it. Others thought that it was a nucleic acid, but they didn't know which nucleic acid it was. So there are three experiments that showed that the nucleic acid carried the cell's information. And those three experiments we're going to take a look at. The first one was by Griffith, and he basically was studying pneumonia bacteria. And so he started working with the pneumonia bacteria, and he found that there were two strands. There was one that he called the R strand because it was a rough, non-virulent, which means that it couldn't cause diseases, that it was inactive, basically. Um, Type, and then there was an S strand, which was called the smooth virulent, and it could kill, it could cause damage or could cause problems for whatever organism it inhabited. So, in his experiment, he took the strain R and he injected it into a mouse, and the mouse stayed healthy. Then he took the smooth or the S strand and he injected it into a mouse, and it died. So then he decided to take the strand that showed that the mouse would die, and he killed it. And so he basically heated that bacteria until it was no longer had that factor that would kill an organism. And so he injected that in the mouse, and the mouse stayed healthy. But then he got a little shocked because he took the S strand and the R strand, and he killed the S strand like he did before. And the R strand, he mixed those together and he injected those both into the mouse. So the R strand didn't cause the mouse to die. The heated S strand didn't cause the mouse to die. But at this point, when he mixed them and he put them into the mouse together, the mouse died. So something had to happen to that live S strand to cause it to come back to where it still had the ability to kill the mouse. So he came up with a couple of conclusions. He basically found that alone the R and S strand did what they were supposed to. The R strand remained harmless, and the S strand caused pneumonia. When he killed the S strand and put it into the mouse, it became harmless. But then he injected the R and S strand, and it caused pneumonia and caused the mouse to die. So his conclusion was that something transferred from the heat killed bacteria to the live bacteria um, was harmless bacteria, making them deadly. So he transformed it. And so this process is called transformation, and it's where one strand of bacteria changes the genes of another strand of bacteria to have a different effect than expected. So Avery, McLeod, and McCarty basically came along after Griffith, and they started looking at this S strand. And so they destroyed parts of the bacteria before combining it with the R strand. And then they sort of did the test to see what had happened. So the destroyed proteins, lipids, carbohydrates, the mouse died. Something had transformed that bacteria. But then he destroyed the nucleic acids, and the mouse lived. So the DNA was the transforming bacteria. It was what was causing that bacteria to act differently than was expected to act. So then Hershey and Chase came along in 1950s, and they worked with a virus that basically only attacks bacteria. It's called a bacteriophage, and it basically looks like those little alien figures that you see in the picture on the right hand bottom side. They use radioactive elements to mark the DNA and to mark the proteins. And then they did their test. They let the bacteriophage inject into the bacteria. They infected the bacteria, in other words. They blended those together to see what would happen when they were blended together. They centrifuged it. And so one side had no sulfur detected in the cells, the other side had phosphorus detected in the cells. So they basically did this whole conclusion was that the genetic material that caused the information to be transferred into what they were going to produce was DNA. So they found that it was the nucleic acid that was basically being the transformer once again. There were also more than one scientist that discovered the structure of DNA, and there were actually four. So there was Erwin Chargaff, there was Rosalind Franklin, there were James Watson, and Francis Crick. Erwin Chargaff in the 1950s started working with the bases that make up the rungs of the ladder of DNA, and he found that there was an interesting mathematical look at this DNA strand. And what he found was that the adenine number and the thymine number were equal, and that the cytosine and the guanine numbers were equal. So, therefore, in DNA, he found that the pairing occurred with adenine pairing with thymine and cytosine pairing with guanine. And this is now known as the Chargaff's rule. So, Rosalind Franklin was the next person who sort of worked with DNA, and she was working with DNA structures in 1952. And she did a lot of photography with x rays, and she found the DNA structure, which is the structure that you see in the left hand corner. Her photo is known as Photo 51. And if you look, it looks like a little X, but that's really how the DNA is curving or making a helix formation. Now, like a lot of scientists in her time, she did not realize that X ray material was harmful to you and that it could cause cancer. And so she did die in 1958 from cancer due to the work that she did with her DNA strand. So Watson and Crick came along and they basically found that by looking at Franklin's photograph of the DNA, he could sort of see how the twisting and curving of the DNA took place. So they basically described DNA as a double helix with strands held together by weak hydrogen bonds, and it formed bases in the middle, which they knew were adenine pairing to thymine and cytosine pairing to guanine. So basically, the DNA structure is going to look like a twisted ladder made up of nucleotides. And remember, the nucleotides are the phosphate group, which in this picture is the little red circles, and the sugar, which is the pentagon blue shape, and then the nitrogen bases, which are the little black rectangles. So the sugars and phosphate makes up the side of the ladder, and then the nitrogen bases make up the rungs of the ladder. Now, when you're looking at this picture, it's flat, it's not twisted like it normally would be in real life. So the atoms within the two strands are held together by covalent bonds, and then the strands are held together by weak hydrogen bonds between the nitrogenous bases. So you have a bond between the two purines. If you had a bond between two purines, you'd have 4 rings. So that would make that area really wide. And if you had it between the two pyrimidines, you would only have 2 rings, so that would make it really small. So your DNA structure wouldn't be very strong because it would be going in and out. So you have adenine bonding with thymine, so you have 3 rings, and then you have guanine bonding with cytosine, so you have 3 rings. And so going up the DNA strand, it basically is very even, very strong because it can be tightly bound together. So, a couple of things that you need to know about DNA. If you have DNA in eukaryotic cells, which is what we've basically been talking about here, it's found in the nucleus because eukaryotes, remember, have membrane bound organelles. If you look at DNA in prokaryotes, there are no membrane bound organelles. So, in prokaryotes, the DNA is found in the cytoplasm. Um, If you were to try to label the parts of the DNA, it would look like this picture. So on the sides, you have all these different nucleotides going up and nucleotides going down. And one of the things that you can look at here is that on the left hand side, the sugars have the pointy part of their pentagon going up. On the right hand side, the pointy part of the pentagon is going down. That's because these always travel from a 5' to a 3' end. And so they do opposites. So they're sort of flipped on each side. And so that helps them to be able to do the coiling for the helix part, and it helps them to be able to be held together. The little dots in the middle between the bases are your hydrogen bonds, and then, of course, the boxes contain your letters that are your nitrogenous bases. And remember that the sugar is deoxyribose. So that's the structure of DNA. And go back through this video until you get the structures and parts in your head, because the next part is where we're going to be replicating DNA. So you're going to want to know about the bases.